CRISPR. Gene editing, the future of medicine. So imagine a world where genetic diseases like sickle cell anemia, cystic fibrosis, or even cancer can be edited out of existence. A world where we can design plants to withstand climate change and create treatments personalized to your DNA. Welcome to the age of CRISPR, the revolutionary gene editing technology that's reshaping the future of medicine and beyond. Hi, I'm your host on Andtops19, where we explore the science and tech defining tomorrow. Today, we'll unlock the mysteries of CRISPR, how it works, its current uses, and the profound ethical questions it raises. Ready to journey into the blueprint of life? Let's dive in. Act 1. The Genesis of CRISPR. Point 1. What is CRISPR? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. Quite the mouthful, I know. In simple terms, it's a tool scientists use to edit DNA with incredible precision. Imagine being able to delete a typo in your genetic code or even rewrite it entirely. That's CRISPR. Think of it as the molecular version of hitting Ctrl+Z to undo a bad decision, like that haircut you got in middle school. Point 2. The origins of CRISPR technology. The story begins in the 1980s when scientists discovered these repeating sequences in bacterial DNA. Fast forward to 2012, Jennifer Doudna and Emmanuel Charpentier developed CRISPR-Cas9, a system that acts like molecular scissors. For this groundbreaking work, they won the Nobel Prize in Chemistry in 2020. But how does it actually work? Let's take a closer look. Act 2. How CRISPR works. Point 3. The CRISPR-Cas9 mechanism. CRISPR is a two-part system. The first part, the Cas9 protein, acts as scissors that cut DNA. The second part is a guide RNA, which directs Cas9 to the exact spot in the DNA sequence that needs editing. Once there, Cas9 snips the DNA, allowing scientists to delete, replace, or modify the sequence. It's like using GPS to find an address and then renovating the house. But what if the GPS got it wrong? Imagine Cas9 editing the wrong gene. Yikes! Don't worry, we'll talk about the risks later. Point 4. Current applications in medicine. CRISPR is already changing lives. In medicine, it's being used to treat genetic disorders like sickle cell anemia and beta thalassemia. Scientists are also exploring CRISPR's potential to target cancer cells, develop treatments for blindness, and even combat viruses like HIV. But it doesn't stop at humans. CRISPR is revolutionizing agriculture too, creating drought-resistant crops and disease-free livestock. The possibilities seem endless. So, what's next for this technology? Could it help us eradicate diseases entirely? Act 3. The future and ethics of CRISPR. Point 5. Potential future breakthroughs. Looking ahead, CRISPR could lead to treatments for diseases we once thought incurable, like Alzheimer's or heart disease. There's even talk of using CRISPR to reverse aging or enhance human capabilities, like increasing muscle strength or intelligence. The future sounds exciting, but it also raises a key question. Where do we draw the line? Should we edit genes to eliminate disease? Sure. But what about giving someone perfect pitch or super eyesight? It's a slippery slope, my friends. Point 6. Ethical dilemmas and risks. With great power comes great responsibility. CRISPR raises serious ethical concerns. For instance, should we allow designer babies, children genetically engineered to have specific traits? There's also the risk of off-target effects, where CRISPR unintentionally edits the wrong part of the DNA, causing unintended consequences. And then there's the fear of inequality. Could gene editing become a luxury only the rich can afford, widening the gap between the haves and have-nots? These are questions we must grapple with as CRISPR advances. Point 7. Regulatory and global perspectives. Different countries have different rules about CRISPR. In China, scientists are already experimenting with human embryos, while in the US and India, regulations are stricter. The global community is still figuring out how to balance innovation with ethical responsibility. So, how do we ensure CRISPR is used for good? The answer lies in collaboration and transparency. Conclusion and call to action. CRISPR is more than just a scientific breakthrough. It's a tool that could redefine what it means to be human. As we venture into this uncharted territory, the choices we make will shape the future of medicine and society. What excites you most about CRISPR? Let me know in the comments below. And don't forget to check out my next video. How do self-driving cars navigate? It's another deep dive into groundbreaking technology. If you enjoyed this exploration of CRISPR, hit like, subscribe to Andtops19 and share this video with your friends. Let's keep the conversation going. Until next time, stay curious and keep exploring.